Welcome everybody who are going to use BioLabDonkey program. In this video I would like to show the functionality of this program for DNA analysis. DNA analysis in this program can be done in three tabs: DNA insert, vector and product. They have similar functions. First we can open, for example, gene bank file. On the upper side you see the graphic representation of DNA features for this DNA sequence. If you move mouse on the top of this arrow, you see the description for this feature. If you click on this arrow, you see the highlighting sequencing in the main window below. Between the graphic representation of the sequence and the main window, you see the several pictograms. If you move mouse on the top of this, you will see the short description. Let's start with the first one. This function is designed to translate, reverse complement and transcribe the selected DNA sequence. For this we have to select the region which we would like to analyze and then press this button, and then automatically we have the transcribe, sequence, reverse complement, DNA and all possible translated open reading frames. If we select, for example, this sequence, then we have parameters for this protein sequence: molecular weight, pi value and length. User have option to save these sequences as text file by using these buttons. Next button is about to search for open reading frames. We have already selected DNA sequence, then we press this button. Here we should first type length of minimal open reading frame. Let's say it's 100 base pairs. Then we have to select the start codon. If we select all, it will use all possible start codons. Then we can change the range of pi value from minimal to maximal value. And then we have to press this button. As you can see, on the upper part, we have the graphic representation of found open reading frames. Green are the forward ones, and the red one reverse open reading frames. Here we have the list of found open reading frames. If we select this, first we see the same sequence in the main window, and second, by selecting this open reading frame, we automatically translate this open reading frame. Here also we have the parameters for this protein sequence. And also we can print, save or discard these open reading frames and start again. After selection, when we press one more time, selection will disappear here and also in the main window. The next function is about search for arbitrary sequence for palindromes, repeats, stem loops, and also if we know start the sequence and end sequence of the sequence, then we also can look if there is such sequence in our sequence. So when we press this button, then we have several options. To find palindromes, we should type minimal length of palindrome and maximal length of palindrome. Let's say it's 7, 20, 6, 20. And then also we can look for inverted repeats. To search for palindromes, we press this button. To search for repeats, we can press this button. Then if we select, for example, this palindrome, the corresponding sequence also will be seen in the main window. Again, if we deselect, will be deselected also in the main window. The same applied to the repeats. And we have two repeats here, and the same for palindromes. We have the position of this palindrome, for example. If you would like to find stem loops, we can also type the parameters for these stem loops, and then we should press this search for stem loops, and then we see the stem loops and positions of these stem loops in the main sequence. If we select, we can see this sequence in the main window. Here, for example, we can search for sequence. Let's say we select here sequence, copy, and again select this region and copy, paste this sequence in search window and then start search. And here we see the position of the found sequence in the main window. We can color this and discolor this sequence. And here you can change the color for found sequence. To search for a sequence which has known start and known end, we have to switch to this function and then type this sequence, and it will and then use this button to find this. Next function is about generation of primers for the selected DNA sequence. So we already selected sequence where we would like to find primers. We press this button, and then I will show you how you can use this function to find the primers for cloning or sequencing. On the left side for ORF cloning, this function is useful, for example, for amplification of open reading frame. And here the function to find primers for qPCR. I will show you how to use the main function for cloning and sequencing. So if you need restriction site, you can type restriction site. And usually you have to have at 5' and several nucleotides for restriction. So let's say we have several T. Then you have to type the length of the primer, let's say 20. Here you can change the GC content, minimum and maximum. Then to find the forward primer, you press this button, and to find the reverse primer, you press this button. We have the several possible primers for this selected DNA sequence. If you select this part before restriction site, you will see this primer in the main window. And let's say you also select the reverse primer, and reverse primer is here. And then if you select forward primer and reverse primer, you can also test these primers. If these primers form dimer, and as you see, dimer is not formed. Besides the search for primers, you can also store the found primers in local database. For this you have to press this button. Then you have the empty line where you can, let's say, copy paste this sequence and give name for this primer. Let's say test primer. And by doing this, you already stored the primer in the local database. So next function is about DNA sequencing chromatogram. Here I have opened GenBank file, but there is option also to open the sequencing file AB1. Let's open this file in another tab. And when I open this, first we see the sequence in the main window, and also we have the sequencing chromatogram. If I select some sequence in the main window, program will automatically move in chromatogram to position corresponding to this sequence. As you see here is the same sequence as we see here. Let's say we can move here. It's also the same sequence. And you can change the width of chromatogram. You can make peaks more narrow, or you can increase the height. At the top of this chromatogram you see the sequence, and when you look at this chromatogram and you see that, for example, here position N actually corresponds to the G, what you can do, you can double click, you can change to G, and then after this, if you would like to save this in original file, you have to press this button, and by pressing this button you will see that you automatically change the N to G in the main window. We can check if original file has these changes. We can close this window, close this file, and again open the same file, and then look at the position. And as we see here, before we had N, and now we have G in this position. So this is about function for chromatogram sequencing. And the next function is about silent removing or insertion of restriction sites in open reading frame sequence, and also about optimization of open reading frames for expression in E. coli or in yeast expression system. About this function and other functions I will talk in the next video. See you!